some structures can't be seen through light microscope. So finer details of plant structure however, can be further investigated. Although widely used in lower plants, electron microscopy has been a comparatively new approach for flowering plants. Micromorphology, or the finer details of external features, have been explored in the recent years by scanning electron microscopy, or SEM, whereas the ultrastructure, or the minute details of cell contents, have been studied through transmission electron microscopy, or the TEM. So, SEM studies have been made primarily on pollen grains, small seeds, trichomes, and surface features of various organs. In most of these organs, the studies involve the epidermis. So, of course, the value of epidermal studies lies in the fact that an epidermis covers almost all the organs and is always present, even in the herbarium specimens. So the epidermis is thick and stable in SEM preparation and is still affected and is little can be affected by the environment. So, however, it is important to note that only comparable epidermis should be studied. For instance, the petals of all plants, leaf of all plants. So, don't compare the petals of some and leaves of others. So, compare petals to petals, leaf to leaves. So, most of SEM studies have been concentrated on seed coats, which is usually thick wall and stable in vacuum, thus facilitating quick preparation for SEM examination without the need for complicated dehydration techniques. So the micromorphology of the epidermis basically the study of the primary sculpture, secondary sculpture, and also the tertiary sculpture. These are some of the SEM seed characteristics of angiosperms. You can pause this video to look at the description, and also you can read the detailed explanation in the textbook. In this slide, it refers to the arrangement of shape of cells. Okay, the arrangement of cells is specific for several taxa. For instance, in Papaveracea, the seed coat cells by a particular arrangement form a reticulate supercellular pattern like this. Okay, like this, this is a family character. So you can use this arrangement of cell to look at to categorize the family. There is specific distribution of long and short cells also over the veins in the family Poaceae. So in the grass family, the shape of cells is mainly determined by the outline of the cells, the boundaries of the walls, the relief, and also the cell wall curvature. Either it can be flat, convex, or concave. So when you talk about secondary sculpture, or we call it a micro relief. This is formed by the deposition of cuticle over the outer wall or due to secondary wall thickenings. So often by the shrinking and collapsing in desiccated cells. It may be smooth, striate, reticulate. Okay, it can be reticulate and also can be striate or micropapillate, or we call it and all members of all members of the urticalis, so urticalis have curved trichomes with silicified cuticular striation at the base. Okay, so this one, okay, they have curved trichomes and have a silicified cuticular striation at the base, and also have micropapillations and on the trichome bodies. You can see that one. So Urticalis is an order of flowering plants consisting of family like cannabis, the Cannabaceae, Moraceae, and Urticaceae. So the single character of trichomes allow the single character of trichomes allows for precise circumscription of disorder orticalis. Orticalis. So this is a special, one special example for this secondary sculpture. So we'll talk about tertiary sculpture. This character is formed by epicuticular secretions such as waxes and other mucilaginous adhesive lipophilic substances, and shows a variety of patterns. So of course, secondary and tertiary sculpturing are mutually exclusive, as the presence of waxes would invariably mask the cuticle. So of course, and then of course the cuticle will be visible only if there are no wax deposits. In monocots orientation, for instance, uh, and and pattern of the orientation and pattern of epicuticular waxes in monocots, four types of wax patterns and crystalloids have been distinguished. These include smooth wax layers, non-oriented wax crystalloids, the Strelitzia wax type, and Convallaria wax type. Okay, so you can refer to the textbook for the detailed explanation. So basically, tertiary sculpture generally lacking from seeds. So for instance, in Orchidaceae, sorry, okay, I repeat again. Tertiary, the seed has been generally lacking the tertiary sculpture, but in the Orchidaceae, certain tribes possess epicuticular waxes on their seed coats. Of course, in Cactaceae, some subfamilies can be identified with seed coat structuring using SEM. Same goes to the Orchidaceae. The family can be characterized using dust seeds, which shows microstructural diversity of the seed coat. Ultrastructure. So studies on ultrastructure have been discerned through transmission electron microscopy, or TEM. As comparison, the resolution power of SEM is 20 times as good as optical microscope, but the SEM is 20 times lesser than TEM. Ultrastructure studies of angiosperms have provided valuable taxonomic information from phloem tissue, mainly sieve tube elements, and then beside this, information has also comes from studies of seeds. So, of course, nearly all angiosperm families have been investigated for the taxonomic significance of these sieve tube plastids. So all sieve element plastids contain starch that differing in number, size, and shape. So the protein accumulates in specific plastids in the form of crystalloids and filaments. So here shows various form of sieve tube plastids and their possible evolution. You can look at this, then you can refer into the textbook for the detailed explanation. Dilated cisternae are the dilated sections of endoplasmic reticulum in the root cells. So originally found in the Brassicaceae and Caparaceae, the dilated cisternae have now been found in several other families of angiosperms, but basically are concentrated in the order Caparalis and form a part of the character syndrome for this order. The dilated cisternae may be atricular, irregular, or vacuole-like in form with filamentous, tubular, or granular contents. Okay, they have been proposed to be functionally associated with glucosinolates and myrosin cells found in this order. The P protein, the phloem proteins, the composition and three-dimensional arrangement of this protein exhibit taxonomic specificity. They are dispersed over the entire cell as the cell matures, but in some dicots, a single non-dispersive or crystalline body of various shapes may be found in addition to dispersive ones. Crystalline bodies are absent in monocots. Their shape is often specific and thus of taxonomic importance. So globular crystalline bodies are found in Malvalis and Urticales. Fabani, which is characterized by PIV subtype plastids, has spindle-shaped crystalline bodies in the family Fabaceae. The features also has supported the transfer of Schwartzia, this genus, to Fabaceae. Nuclear inclusion is another form of protein crystals occur in the phloem and ray parenchyma, primarily in the families of Asteridae. 5 types of crystal has been differentiated. Structural differences are significant for classification within the Scrophulariaceae and Lamiaceae, and also in the Boraginaceae. So protein crystals in sieve tube elements has also been reported and may prove useful. In conclusion, electron microscopy can be the tools to provide further information on microscopic structure for plant classification. The usage of SEM for micromorphology and TEM for uh, ultrastructure will provide in-depth analysis for plant taxonomy.